Hello everyone, welcome. So today we will be discussing about fertilization in plants. We have already discussed about parts of flower. We have discussed about microsporogenesis, that is from microspore mother cells, how it forms the pollen grains. And then we have discussed about megasporogenesis, that is, how from megaspore mother cells it is going to form the embryo sac. And then once this is being formed, the pre-fertilization has already been discussed, or we have discussed how pollination would occur. Now, coming to fertilization of flowers, the first thing which needs to be done is there should be an interaction between the pollen grains and the pistil which is there. So the pollen would come and sit on the pistil, and whether the stigma will permit the pollen to enter into the ovary or not is the question. So if the stigma permits, then well and good, there is an elongation of pollen tube and things proceeds. Otherwise, it says no. The the whole event from pollen deposition on the stigma until the pollen tube enters the ovule, this together is referred to as pollen pistil interaction. In the pollen-pistil interaction, there is a compatibility study of the pollen grains is happening. So once it is compatible from the stigma, the pollen grains from the anther comes onto the stigma. After that, if only if it is compatible, will the pollen tube enter into the style, and then it will reach to the ovule in the ovary at the micropilar end. The pistil has the ability to recognize the pollen whether it is of the right type, that is what I call is compatible, or the wrong type, which is incompatible. So if it is of the right type, the pistil will accept and the pollen promotes the post pollination events that leads to fertilization. The acceptance or rejection is a result of continuous dialogue between the pollen grains and the pistil. The dialogue is mediated by chemical components of the pollen interaction with those of pistil. The compatible pollen grains germinates on the stigma to produce a pollen tube through one of the germ pores. Remember, when we had a germ pore, we told that from there the pollen tube will elongate. So there's a germ pore was a small, tiny pore which was there in the pollen grain, from where the elongation of pollen tube will take place. Next, coming to plant breeding or artificial hybridization. So, for artificial hybridization, as it states, the hybridization or the mix and match is done completely artificially, it is man made. So, what are the various steps I have for artificial hybridization? First, I need to remove the anthers using forceps, and this is called as emasculation. So I will remove the anther before the anther has been matured. Next, I come to bagging, so that there is no other anther which should go ahead and hit the stigma or should sit on the stigma. Hence, I bag it well, and so there is no undesirable pollen grains will which will sit on the stigma. Once the female or the stigma or the ovary gets matured, what I will do is I will dust the desired anther onto the stigma of the flower which was bagged, and then I will again bag and tag it. So I will bag it up and I'll tag it. So what kind of desired thing that took place. So just to note it, emasculation is the first thing. I'm removing the anther, then I'm bagging it. So covering the emasculated flower to prevent contamination, and desired pollen grains are then dusted onto the stigma, and then flowers are bagged again. So for the fertilization occurs in the ovary. Ovary of the flower, as I told, it was inside. You had the stigma, then you had the style, and then inside was the ovary. And inside that was an ovule. So inside the ovary, we said that there's an ovule or embryo sac is there. This female gametophyte, this embryo sac, it has been covered by integuments to to accommodate all the nucellus which is there. Nucellus is nothing but the food storage that is there. So uh, we have, so whenever the entry of pollen tube takes place, the entry of pollen tubes takes place from the micropylar end. Okay, so we have the antipodal cells. Just to give you an overview of the embryo sac, we have the antipodal cells that is at the chelazal end, the three cells which is there, and at the micropylar end we have two synergids and one egg cell, and inside that I have two polar nuclei. So now this fertilization, so what happens when the tube enters, the pollen tube enters, it has one germinative cell and one vegetative cell. The vegetative cell will do nothing, but vegetative cell will direct the germinative cell to move into the egg cell. So, in the embryo sac, we have filiform apparatus which will direct the vegetative cell to come towards the egg cell. Okay, the synergids are having the filiform apparatus. Okay, now once it is before it reaches, what happens? The generative cells gets divided into two cells. So one of them, so when it goes, it will enter into the cytoplasm of a synergid. So once it enters into the cytoplasm of the synergis will degenerate, and the antipodal cells also will degenerate. So one of the sperm will go and it will fertilize the egg cell which is there in the micropylar end. So egg cell along with the sperms will form the zygote, and another germinative cell will go and bind with the two polar nuclei. So, in this two polar nuclei, when it goes and joins, so totally three nucleus is undergoing fertilization. Hence, I called it as a triploid fusion. So, that is a triploid fusion, and here, when in the sperm cells are going to hit the egg cell, there is a syngamy that occurs, and there is a formation of a zygote. So, as it is stated, the process of fusion of male gamete with the female gamete produces a diploid zygote, which is 2N. This is called fertilization. The fusion of one male gamete with the egg and another male gamete with the secondary nucleus is called double fertilization, because I told you there are two fertilizations which is taking place. Hence, I call this as double fertilization. Double fertilization is a characteristic property of angiosperms. It was discovered by S.G. Navaschin in Lilium and Fritillaria species. Now, the pollen tube, after reaching the ovary, It enters the ovule through the micropyle and then enters one of the synergies through the filiform apparatus. The filiform apparatus present in the micropylar part of the synergis guides the entry of pollen tube. After entering one of the synergies, the pollen tube releases two male gametes into the cytoplasm of the synergid. One male gamete moves towards the egg cell and fuses with the nucleus, thus completing syngamy. This results in the formation of a diploid cell, which is called as zygote. This is the number one fertilization that took place. The other male gamete moves towards the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus, that is PEN. And the cell is now called as primary endosperm cell. As this involves the fusion of three haploid nuclei, it is called as triple fusion. Since two types of fusion, that is syngamy and triple fusion, both are taking place, so I call this fertilization as double fertilization. The central cell after triple fusion becomes primary endosperm cell and develops into the endosperm, while the zygote will develop into the embryo. So I told you here, if you see, I have the cells, I have the antipodal cells, the polar nuclei, the egg cells and the synergies. There's the filiform apparatus, the pollen tube will enter, the germinative cells are there. So filiform apparatus is going to guide them, it will go ahead. Then synergists will degenerate and the antipodal cells also will degenerate after fertilization, and there's a formation of primary endosperm cell after triple fusion that is occurring between the male gamete and the two polar nuclei. Hence, I get a primary endosperm cell which is 3N in nature. And also, when the male gamete will come and fertilize the egg cell, I get a zygote which is diploid in nature, that is 2N. Now, what are the various stages of embryo development? So, one, I told you that after triple fusion, the polar nuclei or the cell would get transformed into primary endosperm cell. So, there's an endosperm that is occurring. And once the male gamete comes and fuses with the female zygote, so the egg cell, it will form the embryo. Okay, it will form the zygote. Okay, from the zygote, it will go ahead with the mitosis that will occur. Okay, so this gives rise to the pro embryo that we have, the two-celled pro embryo. So then after that, there is the formation of globular embryo that is there. A globular structure that is formed. Hence, there is a suspensor in the middle, and I have the globular structure. So I call it as the globular embryo, which then forms a heart-shaped. This is the heart-shaped embryo. It's simply based on the shapes, based on various stages that it goes through. Once the embryo develops, then I have the radicle which goes ahead and there is a formation of roots that is there. I have a plumule which goes ahead with a shoot differentiation, and I have the leaf like cotyledons are there. So this is the structure of a mature embryo. So, just to give a gist of what all had taken place, there is lot many things we have made, so many videos regarding this. So, just to give you in short what has happened, we had the flower. From the flower, there was a gynoecium and there was an androecium, that is male part and the female part of the flower. So if I come to the male part of the flower, there was an anther. From inside the anther, if you remember, we had various layers, which is epidermis, endothelium, middle layer, and inside that we had tapetum, which gave the enough amount of food to the microspore mother cell. So this microspore mother cells, it formed microspore tetrad, which among which three degenerated, and there was, each cell it will form, and it will form a pollen grain. So this pollen grain was there. So mm, in the microspore, from the microspore mother cells, we were getting the pollen grains, and this pollen grain was called as microgametophyte, or germinating pollen grain. This would sit on the stigma. So now what happens in the gynoecium, in the ovule? So there is the ovary which is there. Inside that there is an ovule, and inside this there is a megasporocyte, and which is covered by nucellus. So then what happened? This underwent divisions, meiosis. So I have got a tetrad from here. In the megaspore, three cells degenerate because of the calcium content, and I get a complete one gametophyte that is there. And then this megagametophyte, the embryo sac which is there, that forms. There is a double fertilization which occurs. I get primary endosperm cell when there is a triple fusion between the mm, one of the male gametes with the polar nuclei, and then I get the zygote when the male gamete comes and binds to the egg cell. Okay, so once this post for pollinations, once this fertilization would occur, after this I'll get the seeds which is there, and from this seeds again a young sapling would grow, and then I will get the flower, and this cycle will keep repeating. So in a very nutshell, this is what has been occurring in a plant. Thank you.